Welcome to the Immuno Flash Talk event. My name is Jessica Diarra, and I'm an immunology PhD student at Stanford University. Today, we're going to show you several videos that are representing the work of the Immuno Flash Talk competition participants. And we hope that you will be able to watch these videos and assess them, and at the end, vote. Soon we'll just upload the videos so y'all can watch them here.
Juliette, and my project focuses on CD8 T cells that fight pathogen-infected cells such as virus-infected cells, as well as tumor cells, for example. It is well known that for a T cell to get activated, two signals are required, oftentimes provided by an antigen-presenting cell, such as a dendritic cell. Studies have shown that T cells are able to bypass this two-signal activation model when in the presence of a specific combination of homeostatic cytokines and inflammatory cytokines. Therefore, even in the absence of an antigen, these cells can proliferate. This concept is known as cytokine priming. Studies have also shown that cytokine priming can reduce the activation threshold of CD8 T cells. As you can see from the figure, cytokine primed cells have a greater activation level as compared to naive cells, given the optimal concentration of a cognate peptide. The effect is seen when these cells are also given the suboptimal concentration of a peptide. What does this imply? During an immune response, innate immune cells are going to release different cytokines that are going to prime our naive CD8 T cells. These cells are going to get activated and therefore have a heightened antigen sensitivity. Two types of cells can be found in this environment, CD8 T cells that are reactive to foreign antigens, as well as CD8 T cells that are reactive to our own antigens. Upon viral or cancer antigen recognition, this can lead to immunity to infection as well as to anti-tumor immunity. In the case of self-antigen recognition by autoreactive CD8 T cells, this can lead to autoimmunity. My project focuses on the mechanisms involved in this heightened antigen sensitivity. We believe that cytokine priming is going to induce certain changes at the DNA level, which is going to enable a readiness state, so that when they see the antigen, they're able to provide a more robust response. In order to tackle our hypothesis, we looked at the changes happening at the chromatin level using an attack sequencing assay. We found that a significant portion of these modulations were commonly shared between antigen-stimulated cells and cytokine-primed cells. To summarize my project, I'll be using an unbiased approach to compare genes and proteins being expressed by both cytokine-primed and antigen-stimulated cells. I'll then be selecting genes that are relevant to T cell activation. I'll then verify how these genes are expressed in cytokine-primed cells, and if time permits, I'm going to validate these genes using an in vivo model. Thank you.
Hi everyone, my name is Essi Yayra from the University at Buffalo, and today I will tell you about the role of neutrophils in vaccine response to the bacteria Streptococcus pneumoniae. Streptococcus pneumoniae are bacteria that can cause severe disease when they spread to the lungs and other organs. These infections disproportionately affect elderly individuals, as can be seen on this graph, where the case and death rates to these infections increase with age. And this is despite the availability of vaccines on the market. So our main question is, how can we improve the efficacy of these vaccines? And for that, we need to understand the immune cells that are important for making antibodies to induce protection. In a traditional vaccine response, cells known as T cells help cells known as B cells make antibodies that protect the individual. On the other hand, during an infection, the most abundant type of white blood cells, known as neutrophils, fight microbes. And it was thought that neutrophils don't communicate with T and B cells. But we were curious to understand if there's a link between them during vaccination. To ask this question, we turn to a mouse model that mimics humans. When we vaccinate mice with a vaccine against Streptococcus pneumoniae, they make antibodies that protect them against infections. But when we remove neutrophils at the time of vaccination, these mice are unable to mount a protective antibody response, and they die much faster to infection. As you can see here, when we remove these neutrophils, these mice, a quarter of them did not survive. And this is telling us that neutrophils are important during vaccination. We're now investigating how these neutrophils are important. There's a type of T cells that inhibit the immune system, and in data that I don't have time to show you today, we found out that neutrophils inhibit those cells, thus boosting protective antibody production. And with that, I would like to thank my mentor, Dr. Buganem, as well as my entire lab, the NIH for funding this research, and the Black in Immunology Organizing Committee for giving me the opportunity to submit this. Thank you.
We often hear people say, "You are what you eat," and drink too. Many food substances consumed, such as alcohol, can serve as agents of pain and, over time, increase pain sensitivity in humans. Chronic pain affects the lives of over 50 million Americans, with 70% of patients being females and experiencing a greater severity of pain. You are probably wondering why females experience a higher prevalence of pain than males. Now, this is partly due to the differences in the way pain signals are being processed by the nervous and immune system of males versus females. Yes, the immune cells are involved in communication with the nervous system to respond to stimuli. Most communication in this context is facilitated by a special receptor known as toll-like receptor 4, TLR4. Now, TLR4 facilitates the production of molecules that are associated with inflammation and can generate pain for longer periods, resulting in chronic pain. Alcohol has been shown to implicate chronic pain in the context of severe drinking habits. However, we are interested in the other spectrum of pain sensitivity at a lower dose. My PhD project aims to make use of a 5% dose of alcohol over a 14-day period in rodents to mimic low to moderate use of alcohol in humans over a 150-day period. We hypothesize that females would have a much quicker onset of pain sensitivity to a non-pain inducing stimulus than males. Preliminarily, we have noticed that pain sensitivity occurs in females during this short exposure to alcohol and is associated with the activation of immune cells in the spinal cord known as microglia. We also hope to determine whether TLR4 on microglia in the spinal cord is important for pain sensitization outcomes that we see. This work will uncover valuable evidence for male versus female-based mechanisms behind pain development. This work is especially timely because of the possible impacts it can have on the insufficiency of therapeutic solutions that are currently available for pain treatment for male and females. I thank you.
Hello all, my name is Christian Dzuvor, and here is a summary of my immunology-focused research on rational design and evaluation of precision nanoparticle vaccines for beta coronaviruses. Background: the coronavirus family consists of 4 groups: alpha, gamma, delta, and beta. And these coronaviruses have dual in animal reservoir and are poised to spill over into humans. Once more that the beta coronaviruses, which are their threat to human health, have caused various endemics, epidemics, and the recent pandemic caused by the SARS-CoV-2 coronavirus, and has claimed over 6 million lives. New variant keeps emerging and has led to waning immunity. These beta coronaviruses adapt to human using the spike protein, and this spike protein consists of the S2 region and then the S1, which consists of the N-terminal domain and the receptor binding domain, which enable it to bind to a human cell receptor, and has been principal antigens in current vaccines and targets for protective antibodies. Research gap. The research gap behind this project. The current vaccine targets neutralization-sensitive epitopes in the S1 domain, and this S1 domain is subject to escape mutation as new variants emerge in the case of SARS-CoV-2. Also, the lack of sequence conservation within the S1 does not offer broad protection against other coronaviruses. Research overview. Research 621. Rational design spike antigens which could preferentially target conserved protective S2 epitope, as this S2 region is highly conserved across these beta coronaviruses. And secondly, present this new novel designed antigens in different conformation and well-ordered arrays using nanoparticles to enhance and shape a conserved spike S2 epitope immunogenicity. Universal research approach. My overall approach is to compare novel spike nanoparticle immunogen structures, their antigenicity and immunogenicity. And the goal is to identify the best nanoparticle immunogen to take forward into further development. Impact on human immunology. That anticipated impacts on human immunology are: first, develop novel broadly protective vaccine against beta coronaviruses. And second, knowledge gained from the immunogenicity studies will help in further understanding correlate of protection in other coronaviruses. And lastly, to produce certain molecular antibodies and other new assays. And thank you.
Mouse cleaning. I know you're probably wondering why I said that, but how many of you are familiar with the show Tom and Jerry? My favorite episode is called Mouse Cleaning, where Tom Cat's role is to clear the house of Jerry the mouse. Tom is armed with a club to beat Jerry, while Jerry has his own little explosive ways of surviving. For all the years I've watched this show, Tom has never defeated Jerry. In a similar way, malaria has been claiming millions of lives for centuries, with nations incurring billions in cost towards treatment, yet no effective preventive tool has been developed to date. In my experimental design, Tom's house represents our body. Tom represents the immune cells useful for fighting against malaria parasites such as cytokines, B and T cells, while Jerry represents the malaria parasites. In malaria endemic regions of Africa and Asia, there's an element of poverty, thus malnutrition. We therefore hypothesize that low protein diet without zinc and iron micronutrients during pregnancy, as in malaria endemic regions, lowers the adaptive immunity, while a diet rich in protein supplemented with zinc and iron during pregnancy improves the adaptive immune response to malaria. I speculated, therefore, that the possible reason Tom has never defeated Jerry might be because he was born malnourished. But what if Tom's mom was fed on a high-protein diet supplemented with zinc and iron during pregnancy? We could have seen a supercat Tom. To investigate this, we fed pregnant mice on different diets as shown and infected their pups 13 days after they were born with Plasmodium parasites. We observed that all the pups from well-nourished mice controlled the parasites and cleared it, while we lost some of the pups from the malnourished mothers. Additionally, the well-nourished pups had a high number of effector T cells as well as inflammatory cytokines, which are important in controlling malaria parasites as compared to the malnourished mice pups. Ladies and gentlemen, it is interesting how a popular Tom and Jerry tale could tell so much on the effect of moderate malnutrition during pregnancy on neonatal immunity to malaria. And this could be the model solution in malaria research to stop worldwide malaria deaths.
Hello, I know probably you don't like antibiotics. Well, imagine a period where you don't need to use antibiotics to get treated from an infection, such as tuberculosis. Well, my name is Shamsuddeen Yusuf Ma'aruf, and I am a postgraduate student at Stellenbosch University, South Africa, pursuing MSc Molecular Biology. So I'm trying to assess the killing power of nanoparticles against tuberculosis, considering macrophage diversity in an attempt to fight drug resistance, particularly antimicrobial resistance. So just to tell you a bit about tuberculosis is caused by Mycobacterium tuberculosis, which infect the lungs. So in the lungs, we have some military personnel, some diverse military personnel, which we call them macrophage. So this macrophage exists in different forms. Some of these macrophages are meant to repair damages, while some of these macrophages are meant to cause death or maybe kill any foreign substances, such as the mycobacteria. But somehow, these mycobacteria have established a way to avoid this killing effect of this macrophage, which at the end causes a lung disease, which have different events or symptoms such as fever, fatigue, weight loss, night sweats, cough in the blood, persistent cough, and sometimes even leading to death. So attempts have already been developed to fight this TB, such as drug regimens, which include rifampin, isoniazid, paracetamol, ethambutol, which can take up to like 6 months taking this drug. So imagine you in this situation taking antibiotics for like 6 months. Well, the aim of my work is to improve macrophage killing effect considering the diversity of this macrophage. So what we intend to do is to use what we call nanoparticles, which can actually mimic MTB. So these nanoparticles carry what we call immunomodulatory compounds, which are just like weapon-like substances, which are meant to empower the macrophage in trying to kill any foreign substances, such as the MTB. Hence, the objective of my work is to make macrophage a superhero in trying to end tuberculosis. So, me and you can end TB now. Thank you for listening.
Wonderful. Thank you all so much for watching these videos. I quickly want to just share the link to the Flashtalks voting area. So please go ahead and vote. We highly encourage you to please vote. And yes, there it is. It's right here in the chat. Wonderful. Thank you so much. Have a wonderful day. And we hope to see you at all the other Blockade Immuno events.